In this video, we will demonstrate how to isolate a pure colony of bacteria. Until now, we have transferred bacteria from one plate to the next. However, we aren't sure if there is only one species of bacteria in our colony. Bacteria may grow so closely next to one another that they can merge into one colony, giving the appearance that it is a homogeneous collection, when in fact there may be more than one species present. The isolation streak technique allows us to spread the bacteria on the petri plate so thin that we can be assured that one colony consists of one species of bacteria. In summary, a streak plate consists of 3 streaks of bacteria. The first streak will contain the greatest number of bacterial cells, and the bacteria will be growing so closely together that you won't be able to distinguish individual cells. The second streak starts from a location on the plate near the first streak. You will pass the loop through the first streak and pick up bacteria that will be spread to a clear area on the plate. Finally, the third streak will pick up bacteria from the second streak and spread them to a clear area on the plate. By the end of the third streak, you should have individual colonies spaced far apart from one another. We will be using disposable loops. These are sterile. It is imperative that you not allow the sterile end of the loop to touch any surface that is not sterile. If this happens, discard the loop. Pick up a new agar plate. Write your name, date, media type, and incubation temperature on the bottom of the plate. Take the master plate that you previously made. Touch the loop to the colony you wish to isolate. Transfer this colony to the new plate. With the loop touching the agar surface, spread the bacteria back and forth in a region about 1/4 of the size of the plate. Discard this loop. Pick up a clean, sterile loop and touch a part of the new plate near the end of the first streak. You can't see any bacteria, but you should be able to see the markings of the previous loop on the agar. Pass the loop through the area where you previously streaked. This will pick up any bacteria that are on the agar surface. Streak the loop over a clear area about 1/4 the size of the entire plate. Be careful not to go back over the area that you previously streaked. You don't want to pick up any more bacteria as you are streaking. Discard this loop. Finally, pick up a clean, sterile loop. Touch a part of the plate near the end of the second streak where fewer bacteria were previously spread. Pass this loop through the bacteria and spread them over the remainder of the plate. As you do so, it will dilute the bacteria even more. Again, be careful not to go back over an area that already contains bacteria. Replace the lid, turn the plate upside down, and incubate.